Assalamu alaikum. I am Dr. Nur Mohammad Khan, Assistant Professor, Department of Microbiology, Bashundhara Ad-din Medical College. Today I am going to discuss about the viral replication and antiviral drug. So, in case of replication, the virus replicate in different way in contrast to bacteria and other eukaryotic cell. As virus only contain its nucleic acid and other proteins such as capsid protein or virion specific polymerase enzyme, it replicates one virus may produce hundreds of progeny virus in the process of replication. And this replicative stage can be divided into three main events, that is early event, middle event and the late event. An early event consists of attachment, penetration and uncoating of the viral genome, and this attachment and penetration is have got specificity between the virus and the host cell and in specific receptor interaction with the virus particle. Then the middle event consists of messenger RNA synthesis as well as genomic replication. The messenger RNA synthesis is divided in two phases, that is early viral messenger RNA and late viral messenger RNA. The messenger RNA which divide before which produce the messenger RNA which produce before the genomic replication is called the early messenger RNA, and those produced after the genomic replication of the virus is called late messenger RNA. And last of all, the progeny virion assembly and release from the host cell. Now, the attachment, penetration and uncoating, it is specific for this attachment is specific for the virus and the host cell specificity, and it is mediated by attachment of the viral specific protein such as capsid protein or glycoprotein with the interaction with host cell. Different type of receptor is used by the different viruses group, such as human herpes virus, it use fibroblast growth factor as its receptor, and in case of HIV virus it uses the CD4 protein of T lymphocyte as its receptor. Then attachment and uncoating, and uncoating means the virus the viral nucleic acid which is covered by the capsid protein and consists of nucleic acid. This capsid does not enter into the cytoplasm, may enter or may not enter, only the nucleic acid is injected into the host cell, and this process is called the uncoating. Then the middle stage also called the gene expression and genomic replication, and in case of gene expression, first of all the synthesis of early messenger RNA from which the virus produce structure the virus produce enzymatic protein that are necessary for its genomic replication, and after genomic replication it produces late messenger RNA which usually produce after translation the structural protein, and with this structural protein progeny virions are formed and released from the host cell. Now, this gene expression and genomic replication is different in DNA virus and RNA virus. In case of DNA virus, this genomic replication occurs into the nucleus, and there is some exception, such as in case of pox, although it is DNA virus but replicate into the cytoplasm, and the RNA virus which usually replicates into the cytoplasm, but there is also exception such as retrovirus which replicates into the nucleus as well as the influenza virus. Now, we know all the DNA virus got double stranded genome except the parvovirus, and all the RNA virus have got single stranded genome, RNA genome, except the rotavirus. And There is another exception in case of retrovirus, which have got diploid genome. Others all genome are haploid. Now, in case of DNA virus, I have already mentioned that the replication occurs into the nucleus except pox virus, and the double strand acts as in the negative strand acts as in acts the negative strands from the negative strands the messenger RNA will produce and also produce the genome for its replication, and its accident the negative strands acts as a template. And in case of RNA virus, there is some sorts of difference among these RNA virus group. We can classify it into 4 categories, such as some RNA virus have got single strand positive polarity, and some got single strand negative polarity, and one family got double strand RNA genome, and this variation also causes difference into the replication of this virus family, such as if you consider the first group, those group which contain single strand RNA genome positive polarity, such as poliovirus and other enterovirus. This single strand RNA genome acts as a template, and there is no need of virus specific polymerase enzyme, and this messenger this RNA can be act as a template, and the host cell polymerase can produce messenger RNA from this genome. It included the poliovirus and as well as other enterovirus such as hepatitis A virus and rhinovirus family. Those contain are positive strand RNA. Now, the second group, these are single stranded but negative polarity. Those RNA virus must have virion specific RNA polymerase, as these negative strand cannot be used as a template for the synthesis of messenger RNA that are used to produce enzyme and as well as genome that as well as the second group of virus that have got single stranded RNA with negative polarity, such as measles virus, measles, mumps, rabies virus, and orthomyxovirus family such as influenza virus. And in this case, virus must have RNA dependent RNA polymerase to produce positive strand from the negative strand genome, as negative strand genome cannot be used as a template for genomic replication. And the third group, which contain the double stranded RNA such as the Ryu virus, and this shall have no enzyme to transcribe RNA to messenger RNA and must contain the virion specific polymerase. And the fourth group, such as retrovirus, its polarity, genomic polarity is positive polarity, and its genome is diploid, but RNA is single stranded, and it contains reverse transcriptase to transcribe this RNA into a complementary DNA, but after that this intermediate DNA can be transcribed into viral messenger RNA with the host cell specific RNA polymerase. Now, the last step, we know the assembly and the release. In this case, in this event, the progeny that are formed into the host cell, the genome and the structural protein, capsid protein are assembled and packing occur, and this nucleocapsid, newly formed virion, can release into 2 ways. One is the process of budding. In this case, it contains envelope from the cellular membrane of the host cell, such as in this step followed by the every envelope virus, and there is an exception in case of herpes virus, which got its envelope from the nucleus, and budding occurred through the nuclear membrane, and there may be lysis may occur. Lysis may occur in case of non-enveloped virus, the host cell lysis and the newly formed progeny virion released and can affect the another cell and repeat this cycle again. So, this is in short about the viral replication. Thank you.